Good evening once more, and as I indicated, today's lecture is going to focus on a fermenter and the fermentation process. Here we're going to describe the fermenter, which is the vessel with control environment used in fermentation. And we'll look at the fermentation process, basically having a broader picture on the different stages that are employed in the fermentation industry, from upstream processing through the fermentation itself and downstream processing. The purchase of raw materials, the selection of the right inoculum, the regulation of the conditions within the fermenter, and of course the harvesting process, the purification process, the packaging process, and also the marketing process. That's the global picture of what I'm describing as fermentation process. So we already saw this diagram in our lecture on metabolic methods, and we agreed in that lecture that whatever path, whether we're using the Enden-Meyerhof partners pathway, whether we're using the Edna-Jodorov pathway, Pentos pathway, pathway to get usually to get to the pyruvate, the sole goal is to obtain pyruvic acid, from where used as the starting material for the production of our desired product. So again, just to rush over, we saw that if our starting our fermenting organism is preparing bacteria, the preparing bacteria, then our product is going to be cheese. Our fermented product will be cheese. If we're using aspergillus species, lactobacilli and streptococci, then we're likely going to get yogurt, we're going to get cheese, we're going to get cheddar from lactic acid. If we're using the yeast Saccharomyces from alcohol, fermenting alcohol, we're likely to get wine and beer. And the list continues, as you can easily read. As I indicated, this is something we have gone through already. So my interest today is to look at the industrial process itself. I already started saying that a fermenter is a vessel which has been designed for the growth of microorganisms under controlled conditions. It is also designed for the production of desired byproducts. So could either be interested in microorganisms, that is biomass, or the product of the organism. It could be secondary metabolites, it could be primary metabolites. And these products could be intracellular or extracellular end products. So whatever the case, we'll be looking detailedly shortly. Under control conditions, we can stimulate the organism to either grow in excess if you are interested in biomass, or to produce primary metabolites in excess, or to produce secondary metabolites in excess. Some of these conditions will include also adjusting the parameter of the fermenter to give the organism optimum growth environment by adjusting the pH, adjusting the temperature, providing appropriate gas, that is either oxygen, or making sure we don't have oxygen, depending on whether it's we're dealing with an aerobe or an anaerobe. We want to make sure that the heat does not build up in the system while the fermentation process is going on, because if temperatures go so high, it will affect growth of the organism. We also want to agitate to ensure uniform mixing and to get rid of foam that is usually formed as a result of high protein synthesis or as a result of stirring. I mean, the agitation process itself. So I try to have two pictures here, one being the typical fermenter in the industry, and then a schematic of the fermenter to let you understand the different components of the fermenter so that you'll be able to appreciate what you see in the industry. Remember, it is also called a bioreactor. So we either use the word fermenter or a bioreactor. It's or something. So let's start by understanding the schematic diagram. At the very top of the fermenter, right top here, we are going to see what we have what we call a motor. A motor is to ensure rotation, free rotation, and to the motor is attached a foam breaker to which when the motor rotates, it breaks foam that is forming at the top of this fermenter. Secondly, is also attached a flat-bladed impeller. This flat-bladed impeller is used for stirring. So when the motor rotates, the impeller stirs of the medium, either distributing temperature evenly, distributing nutrients that have been introduced evenly, distributing oxygen and other gases evenly, and so on. Of course, as I indicated, this agitation process usually generates foam, which is also again broken down by the foam breaker attached here. The fermenter is insulated with a baffle. The baffle is meant to insulate the fermenter so that there is no direct heat exchange between the external environment and the fermenting interior environment. Then, of course, we introduce our culture broth into this process. The culture broth will also contain our substrate on which the microorganism will grow, fermenting this substrate or converting this substrate to either biomass or primary metabolites or secondary metabolites. Then we also have a cooling jacket. This cooling jacket is meant to cool and maintain adequate temperature for the fermenting interior. And there is steam sterilization, which is applied because we want everything that is being fermented to be done by our selected fermenter. You saw that if we don't have Saccharomyces cerevisiae, for instance, we're not going to get beer. If we have another fermenting organism, we are going to get a very different end product, which might not even be alcohol. So it's important that we don't contaminate this system so that we have the desired end product. Of course, there's also an acid-base controller system because the fermentation process results in the microorganism excreting waste, and these wastes are bound to build a change in pH. So we need to bring the pH back to normal so that the organisms can continue to grow. If you look at the other diagram, you see the fermenter there, well designed, and if you pass through a Brazilian brewing point either in Yaoundé or Douala, even in Bafoussam, you're going to see this typical structure there, and with all these pipes connected. Some are inlets, some are outlets, depending on what is being done there. Okay, that is the fermenter. That is where fermentation takes place. The diagram we just saw is where fermentation takes place. But before fermentation, a lot of activities are going on. A lot of activities are going on. The first one is what we call supplies and outbound logistics. Supply and outbound logistics. So we are looking at things that we have to do before we begin the fermentation process. For instance, we have to make sure that we have the raw materials that we need for our fermentation. We also need to have adequate storage facilities for these raw materials. Some of them might depreciate so fast, some of them may not need water, some of them may need some primary processing before they are stored, and so on. So we need to have all of this in mind before we begin the fermentation. After we have them, we need to underdo what we call upstream processing. Upstream processing, I will talk in greater detail shortly, But in summary, we'll have to deionize, we'll have to pasteurize, we'll have to blend. Of course, this is not done for every process or every fermentation procedure. It depends on what you want to do. Okay, this is a schematic. From the next class, I'll start treating each industrial product. So we'll start with beer, we'll go to dairy products, we'll go to vaccines and stuffs and antibiotics. So this is a schematic of the entire process. In some cases, for instance, in brewing, we'll not do ionization, but we'll pasteurize. Pasteurization typically in brewing will instead come at the level of downstream processing. Blending, of course, we'll have to blend the math. So we'll this is just a schematic. We'll get to the detail for the different specific processes. After we've done this preliminary processing, we now introduce the organism, this organism, that's a fermenting organism, plus the substrate into the fermenter, regulate the conditions of the fermenter, and then set it on to run the fermentation process. When it is done, we have to harvest and do what we call downstream processing. We may have to undertake precipitation, again deionized, we might separate by chromatography, we might separate by evaporation, we may filter, we may crystallize, or we may dry. It all depends on which product and how the product is being formed. After that, we do what we call outbound logistics, which will include us packaging the product into, branded forms that can easily be marketed, and then now other store will deliver them to customers. There's one of you with your microphone and another device on at the same time that is bringing me echoes. I think it's just taken care of now. Good. So I've just again highlighted the fermentation process here is in words. As we start with inbound logistics, which will include delivery and storage and maintenance of raw materials. We have upstream processing, which will include sterilization, pasteurization, deionization and stuff, depending on what we have and what we need to do. Then we have the fermentation process itself, by adjusting the fermenter conditions and the environmental conditions. And then we'll have downstream processing and then outbound logistics. I've already explained all of this on the diagram. But again, let's go into greater detail to look at the different stages, treating inbound, upstream, downstream process and upstream process separately, and going into greater detail, what are we doing at each of these processes. But one of the most important things we need to note is that before every process can be initiated, we must be able to first select and propagate our fermenter. I'm talking here about the organism. We must be able to isolate and propagate the organism that we use in fermentation. If we cannot have the right organism, we will not have the right products. That's why in the second slide of this lecture, I took time to show you that if we are having Saccharomyces cerevisiae, for instance, our product is going to be wine or beer. If we are having lactobacillus or streptococcus, our product is going to be yogurt. So it is very important that you have the right organism and the right strain. It's a whole laborious process on its own, requiring a lot of procedures. But I'm sure from your general microbiology lecture and other lectures, including with me, we know how to obtain and isolate. Each and every one of you here can obtain and isolate from either the field or from stool sample or from any waste material. You can go to culture and then obtain and isolate, and then subsequently obtain a pure culture. When you get this pure culture now, the next one is this pure culture the right organism? Because we might need a strain, a particular strain of that organism. Maybe not every species of that organism is able to undertake that fermentation. So we might need to identify the right strain that will be able to ferment our system. The next worry could be that the right strain has limitations. One of these limitations could be that it cannot tolerate high acid concentration. Another limitation could be that it cannot tolerate certain temperatures which are optimum for the fermentation process. There are so many of these that I can highlight. Of course, I'll be talking about some of this again uh, as we move on. So, whatever the case, we may have to improve on the strain. Okay, in one of my lectures, I'll be teaching you what we call strain improvement. There are so many methods. It could be by direct mutation, it could be by genetic engineering, and so on. So we have to improve on this strain to either make it tolerate temperature, to make it tolerate a high concentration of the end product, to either make it increase the yield of the product or of the desired product. So there's a lot we need to do that. Let's look at the typical stages again with more emphasis on what is being required at what stage. So let's begin with upstream processing. In upstream processing, there are two things we want to look at here. The first one is our inoculum. The second one is the raw material, that is the substrate. Okay. Beginning with the inoculum, as I was already saying, we need to be able to isolate the organism. When we isolate the organism, we check if the strain that we have is the desired strain, that is withstanding the fermentation temperature, fermentation oxygen, end product concentration, and maximizing product yield. When we confirm all of this, we can now produce this strain in higher concentrations. We can produce these strains in higher concentration. So we do a good quantity of it, and then we save in small vials. I'm sure you can remember again the preservation, the lecture on the preservation of microbes. So when we record that lecture, we have to preserve in small vials, some at very, very low temperatures of minus 80 degrees. We might even have to lyophilize some. And then we have what we call the using stock, the running stock, which is what we use for our daily experimentation or daily production until that stock is running out before we go back to our major stock. Why do we need to do this? Remember that microbial isolation is a very cost and labor intensive process, but more intensive in strain improvement. If we have to engineer a strain every time we want to isolate an organism for our production, then most of our costs will be going in for that. And then secondly, we are not sure that every time we always have it right. Okay, it is a chance process. We try to get genes engineered or get a mutation introduced by chance. Of course, we have a higher degree of certainty, but it's not always sure that everything will go right. So we want that this strain that we've improved and tested to be producing the desired products should be preserved so that for generations we can be able to comfortably use that strain. One of the things that is also tested during this strain improvement and production process is the nutritional requirements of the strain. For instance, if the organism requires nutrients that are expensive, if it is feeding on nutrients like fossils that are also used by the automobile industries and other major industries and driving engines, it becomes a very competitive process to obtain this substrate, meaning that what? The price of the end product will be very high. If products are acquired at a high price, the price of the end product will also be very high. That will not be competitive enough in the market. That won't be competitive enough in the market. So we will have to do something in strain improvement to adjust that or totally disregard that organism and go for another one. The next thing we want to look at is temperature optima. We know that our fermentation process will go up to a particular temperature. So if that organism cannot survive that temperature, we'll either have to improve it to survive it, or we just have to change it. There's no way you use a fermenting organism that cannot withstand the temperature of the fermentation process. Okay, so we'll also look at the oxygen and carbon dioxide requirements of the organism, shear sensitivity. How can the organism withstand the agitation and the stirring that will be done during the process? The genetic stability of the organism. Remember, I said we have to keep stock, we have to subculture, and then we now use for fermentation. If the genetic makeup of the organism changes so fast, if the organism is so susceptible to mutation, it will mean that within a very short time, we are going to be having a different strain, and that strain might not even be yielding the desired end product. So we are going to have to always culture or subculture, isolate and improve more often, and it is not economically viable. It's not competitively stable in terms of price stabilization in the market. Yes, who is helping me with muting? Just be very swift with that. I appreciate that. Thank you. Okay, that is it with the strain. When we have our strain, we now come back again to the raw materials. The raw materials, we're actually looking at the substrate on which the organism will either fit and grow and multiply, or the substrate that will be used to ferment and produce our desired end product. So when we go to the brewing process, you take a particular note that we are using a substrate for alcohol and another substrate to feed the organism. So there are two different things in some cases. In some cases, it's one. But the main things we are considering here is what? The stability of the substrate. If it is a substrate that is not stable, it will mean that anytime we buy a small quantity, we have to use, or else it will going to get bad. That's the problem, especially if the substrate is not produced nearby. For instance, malt, which is used in the production of beer, in Cameroon is all imported. So if you cannot preserve the malt for a certain length of time, you have to import every time, then it is not going to be competitively stable for you in the market. We also want to ensure that the substrate is able to provide the common sources or the main sources of nutrients to the organism and also trace elements. The substrates are able to provide carbon, nitrogen, phosphorus, and sulfur, and also tracer elements as we saw in one of our lectures last year. Then does the substrate require a lot of pre-treatment, which will be associated with cost? Do we need to always sterilize with a high cost before we can be able to use our substrate? Is our substrate readily available? Do we need to always import everything, or do we need to buy just around? Is it competitive for other products? For instance, is it used for other products? I used the example of fossils earlier. We can also use the example of maize. If maize is being consumed by humans and by microorganisms, then you see that the price of maize is relatively higher and might affect cost. Of course, we'll be looking at some of these things when we're when we're understanding the process of brewing. But a more reliable and cheap substrate, which is usually a waste for humans, and a good substrate for the organism is ideal, as the price will usually be very, very, very cheap. So when we have our substrate, as I already started indicating, we might have a different substrate for the maintenance of our media and a different substrate for the fermentation process. Okay, so know that there could be a substrate for our culture inoculum and a substrate for fermentation. And in some cases, it might be the same. Then when we have all of this, the substrate plus the organism, we fit it into the fermenter, we adjust oxygen, we adjust pH, we make sure that this foam generated is taken care of, we ensure that cooling and temperature is well regulated, then we could switch on the process and hope to harvest our end products. After fermentation, the next task, which is also very important, is what we call downstream processing. I already started indicating to you what downstream processing was, but we need to know that it is influenced by byproduct concentration and stability. Okay, the byproducts could be our desired products. In some cases, the byproducts are not our desired products. They use at each step the cost of purifying these end products. I'll call end product because they could be byproduct or they could be the biomass, are all things that come into play in determining the right procedure for the fermentation cost and the right procedure for downstream processing. So when you look at this schematic, I want to gradually explain it so that you understand it as much as you can. There are three different stages of downstream processing that we are going to understand. The first stage is what we call primary recovery. The second stage is what we call product purification. And the third stage is finishing the product. That is the first thing to know about downstream processing. There are three stages that we'll be employing. Primary stage, which is the recovery, the product purification stage, and then the finishing procedure to send it to the market. Now, the major factor that will decide which procedure we employ in downstream processing is one, whether we are interested in biomass or we are interested in microbial byproducts. Let me take it over. The major decision that will decide, that will use to influence which technique are we going to use in downstream processing will be either whether we are interested in biomass or we are interested in byproducts. The next decision is if we are interested in end products, is the end product of interest intracellular or extracellular? So look at this arrow. Our end product could be extracellular or our end product could be intracellular. What do I mean here? The organism grows and secretes these end products, but this end product stays either in the cytoplasm of the organism or the end products are excreted outside of the organism, which means that they will be found in spent medium. If they are excreted, if they are extracellular, they will be found in spent medium. But if they are not excreted, it means that the end product will be found inside the cell. So if the end product is inside the cell, we are going to harvest it just as we harvest biomass. Now, since our end product is not biomass, but an intracellular product, we need to disrupt the cell. We need to break down the cell. When we break down the cell, we are expecting that our end product now is going to be released. When the end product is released, we can centrifuge, remove the debris, and then have the end products ready for secondary purification, what we call product purification. On the other hand, if our end product was extracellular, the first thing we need to do is what? Separate biomass from the spent medium. And second, we extract the end product from the spent medium by concentration techniques. After extracting the end product through concentration techniques, it will now be ready for what? Product purification. We'll go to the secondary stage now. In this product purification stage, depending on the end products, I mean the solubility, now we're no longer looking at whether it was external or internal, extracellular or intracellular. It's no longer an issue here. We all have it now in the same liquid. So how do we separate the end product now from that liquid? We could use dialysis, we use precipitation, we use partitioning, chromatography, ultrafiltration, distillation, and so on. Any of these techniques are employed to separate the end product from the solution in which they are formed. And when this separation is done, we now go through the finishing process by crystallizing the end product, by drying it, by lyophilizing, sterile filtration, or packaging. And when that is ready, we are ready for the market. Again, if you have a question, feel free to interrupt and ask, or you could write it down at the end of the recording. You could come up with a question and we discuss. We have now understood the schematics of the fermentation processes industry, which begins with inbound logistics and downstream processing through fermentation to the upstream processing and outbound logistics. But let's now concentrate in the middle part of it, the fermentation process itself. There are two different types of fermentation. We have what we call liquid phase fermentation and solid phase fermentation. In liquid phase fermentation, the fermentation process is undertaken within an aqueous medium. So the cells and the substrate are suspended in an aqueous medium, and the fermentation process goes through, and then the products are harvested and processed in what we just described in upstream in downstream processing. In solid fermentation, on the other hand, we have very minimal liquid present in our medium, and therefore the organism, the fermenting organism, is absorbed onto the solid solid substrate, which also serves as nutrient material for an organism. And as organism is feeding on this substrate, it is metabolizing it and converting it to the desired product. Liquid fermentation can be divided into three different steps. The first one is what we call batch fermentation. In describing the typical microbial growth curve, we have been able to understand what we call a batch fermentation. So here we put everything together, the inoculum, the substrate, adjust temperature and other environmental conditions, and then we close the vessel. We don't add or change any of these conditions, allow it to run to the end, and then we harvest. That is batch fermentation. Another type of liquid fermentation is what we call fed batch fermentation. In this system, we set up just as we did with a batch fermentation, but there is a difference. We could add one or two more components, especially essential nutrients, periodically, to ensure that the fermentation process is maintained at the logarithmic phase in most cases. So if our product of interest is a primary metabolite or biomass, we are going to ensure that this phase is maintained so that the fermentation process continues at a logarithmic rate, and the end products are also produced at a logarithmic rate. But if our product of interest is a secondary metabolite, remember I told you that secondary metabolites are produced at this stage of the growth cycle. So we'll adjust the required nutrient and maintain this stage of the growth cycle so that the secondary metabolite is excessively produced at a longer time duration. Semi-continuous fermentation is also similar to fed batch fermentation, but the difference here is that we adjust a lot more than in fed batch fermentation. So we could estimate at midway into the fermentation process, we harvest and replenish the medium and even the starter culture. Okay, so in fed batch, we're adjusting just one or two nutrients to maintain the logarithmic phase or the desired phase of the production process. But in semi-continuous, we change both the nutrients, the spent nutrients, and the inoculum. We adjust both the nutrients, the fresh nutrient and spent nutrient. I will also adjust the inoculum and the products. So we could be harvesting and removing waste, and also at the same time adding fresh nutrient and also fresh inoculum. Again, we could want to maintain a particular pace of the growth cycle. Continuous fermentation is an extension or an elaboration of semi-continuous fermentation. In this case, we continuously supply, unlike in semi-continuous where we will halt at one point, remove this and add this. In semi-continuous, we are continuously adding fresh nutrient. We are continuously adding nutrients. I'm sorry, the starter culture. We are continuously harvesting end products. We are continuously removing waste from the medium. All these parameters have been well calculated to know exactly the flow rate, the rate at which we are adding, and the rate at which we are harvesting. So this goes on until the process, there's no end to it. That's what causes continuous fermentation, and is the most widely used method in fermentation in most industrial processes. We've just looked at liquid fermentation. Let's now look at solid state fermentation. Solid state fermentation, I want you to go back to practical realities. Take water fufu, for instance. If any of you have produced water fufu, the process is carried out through what we call solid state fermentation. Remember, we said that there's minimal water. The organism is adhering or absorbed by the substrate, and the organism ferments the substrate, and then the substrate is ready for harvesting. In most solid state fermentation processes, a paste is made up of the liquid, of the medium. So it could be wheat, it could be grains, it could be maize, it could be cereal, it could be rice. So we grind it and produce a paste out of it. When we produce this paste, typically, like you do with water fufu, you grind cassava and you produce a paste out of the cassava. When you have this paste, you can now moisten it a bit. It depends if there's not enough water, but if there's no water, you don't need to add more water. The paste is enough. You now add the inoculum. The inoculum now is your desired selected microorganism, the selected starter culture, the selected fermenter. When we add this microorganism into it, we could want to supplement the medium by adding some salts, some other micronutrients or minor elements. When we add all of this, we now allow it to stand and ferment for some time. Of course, making sure that we have appropriate temperature and other environmental conditions at optimum level. When we have all of this at the end, we harvest, and then our product is ready for downstream processing and packaging. Again, when I want to talk about solid state fermentation, I always want you to look at your water fufu, which you usually prepare and eat. That is a typical example of solid state fermentation. The only difference there is that you don't add an inoculum, you don't add a microorganism, you also don't add any supplements. Now, what then is fermenting your water fufu? Your water fufu is fermented by unknown organisms. Okay, you just assume that the cassava, by the processing procedure, is contaminated by some fermenting organisms, and when you allow it to stand for some time, the fermenting organisms ferment the cassava and then transform it into water fufu. That is why water fufu can never be uniform in taste. If you taste certain water fufu is sour, some are not sour, some are, it cannot be uniform because you have not sterilized the cassava and you have not selected a particular fermenting organism for that process to be carried out. So I will implore you to read the remaining section of this note. I've already highlighted them already. I've already mentioned what you control here. The most important thing is that you should know that these things are regulated, these things are controlled to optimum for the organism. I'm not interested in you mastering the details. That's why I'm not explaining it. You don't need to master the details of all these oxygen stuff, pH, temperature, and raw materials. You don't need to master all these details, but for raw materials, you need to be able to tell me what you should consider in choosing the right raw material. I said already, you need to look at the cost, you need to look at the stability of the raw material. You need to look at the cost. This is cost and availability. You need to look at the ease of handling, ease of transportation, ease of storing, sterilization requirements, and pre-treatment requirements. You need to look at formulation and mixing. You look at the concentration of product produced. That is, how much yield can we obtain from it? If the yield is very low, then the cost will be very high. It's not a good product. You also look at the level of impurities that are there and the safety considerations of discharging the spent medium. So if you cannot safely discharge the spent medium into the environment, then there's a problem. Same with the microorganism. If you cannot safely discharge it into the environment, then it becomes a problem. There is additional cost with that and a health concern, which could, disturb our handling of that. With that, I will stop the